All right, we're just about finished with today's video. I just wanted to go over a couple of slides about cancer. It's not super high yield on the MCAT, but you definitely should know it because it does pop up. And again, a lot of times you'll see it in the context of a passage or something like that, but it's always good to have background knowledge. Okay, so oncogenes and tumor suppressors. Cancer is an inappropriate cell division that may be present as a tumor or in a more diffuse state, such as leukemia. In the most basic sense, that's really what it is. It's just inappropriate cell division. Something went wrong, something went haywire somewhere, so now cells are dividing unchecked, and that leads to tumors, and eventually it could lead to death. So a mutated gene that induces cancer is referred to as an oncogene. Oncogenes are actually genes that are normally important for cell growth and maintenance, but have been mutated. So for example, from UV rays or from chemical mutagens, and they turn malignant. So these are genes that everybody has. They are important for normal functioning, but they're specific genes that are more likely to become oncogenes than others. And a normally functioning gene is called a proto-oncogene before it is converted into an oncogene. It's just the nomenclature that's used. So every cell in your body is also equipped with tumor suppressor genes that code for proteins that survey the cell for abnormal growth and initiate apoptosis if necessary. The protein P53, for example, is a product of a tumor suppressor gene that you may come across on the MCAT. And P53 is actually pretty important in surveying the cell and activating apoptosis if there's no possible way that the cell can be repaired. So high levels of P53 in a cell will activate apoptosis. So apoptosis is a form of programmed cell death that's employed by the cell when there's no chance for repair. It's quiet, it's controlled, and it seeks to minimize damage to the surrounding cells. So that's why we say it's programmed. If you compare it to other types of cell death, say like necrosis or lysis, it's not as quiet and it's not as safe for the surrounding cells. So it can cause some damage to the tissue. Apoptosis, while it is cell death and it's a last resort, it is quiet, it is controlled, and it keeps the surrounding cells relatively safe. So the cell begins to shrink during apoptosis and the cytoskeleton is disassembled and the nuclear envelope is broken down. So the genome becomes fragmented throughout the cytoplasm and the shrinking cell exposes new proteins on its outside to signal to phagocytic cells to finish its degradation. So it sort of just becomes like a soup inside. It loses the cytoskeleton, it loses its structure, and it sort of starts, it undergoes this process that's known as blebbing. It sort of turns into these small little blebs that pop out, and eventually it forms these like little tiny bodies that we call apoptotic bodies. And those, like we said, are going to express some weird protein profiles on the outside, so the phagocytic cells can find them and finish degradation. And then, recycle anything that it can recycle and get rid of anything that it can't. Degradation inside the cell during apoptosis is carried out by enzymes known as caspases. So they have a cysteine residue in their active site, and they cleave proteins at aspartic acid sites. So that's actually where the name came from, C as in cysteine, and then ASP as in aspartic acid. So caspases are inactive until initiator caspases receive internal or external death signals. So initiator caspases are the first line that become activated during apoptosis, and they can then activate more initiator caspases and then effector caspases. So those are the ones that cleave cellular proteins and they trigger apoptosis.